All right, so it is 2 o'clock. We will get started. And I just realized I did not plug in my tablet, so just give me a sec for that. All right, there we go. So let me pull up the PowerPoint. So we just have a little bit to finish for Monday's lecture. Okay. All right. So
On the whole trial.
oh, one sec.
Yeah. We'll talk about it later. I'm gonna go get some grub. I'm gonna be downstairs.
There we go. All right. So we ended up talking about bases, DNA, how DNA is coming together, 5' to 3', how the phosphate connects to the sugar, how the sugar connects to the base, and all that good stuff. And then we talked a little bit about the biochemistry behind our double-stranded DNA, why they can be separated at basic pHs, and why increasing salt concentration will make DNA harder to melt. Last thing we just need to talk about for DNA for this slideshow, and then we'll go on to the next one, is just how DNA replicates. And it's just going to be a very quick overview. The first thing we want to start with is just the overall central dogma here. DNA itself can be replicated to make more DNA, or DNA can go through transcription to make RNA. This RNA can be translated to make protein. And so that is the central dogma of biology right there. Now, DNA itself is called semi-conservative because when DNA replicates, as shown in this image, one strand is always the old parent strand, and one strand is the new daughter strand. So DNA is always semi-conservative, and DNA can do this because of the base pairs. A is always with T, G is always with C. So if you know one strand, you know what the other strand must be. Now, just going back to the central dogma, what's being shown on this far right here, when DNA is transcribed into mRNA. These mRNAs are read in triplets, which are called codons, and they're matched up to a tRNA. These tRNAs are what are carrying our amino acids, and these amino acids are put together at the ribosome to make our protein. And we'll go into that a little bit later on in the semester, look at some of the biochemistry of that a little bit. So just so we understand the whole A to D, G to C business, I'm sure we probably do. I just want to make sure and give us a little practice. Let's say we have a diploid, and this diploid organism has a 45,000 haploid genome. This contains 21% G residues. I want you to tell me, based on that information, how much A, C, G, and T are in each cell. So we're just going to begin with that quick calculation here. All right, so while you're working on that, I'm just going to write down some basic information that we can draw from this problem. So we have well, let's just start with the amount of G, AC, G, and T percentages. So G, we're giving 21%. From Chargaff, we know that the amount of G has to equal the amount of C. So the C in the organism has to also be 21% because G base pairs with C. So overall, that makes up 42% of our genome. Therefore, the other 48% must be A and T. And since the amount of A equals the amount of T, that means A is 24% and T is 24% as well. So that's how we can use Chargaff's rule to determine the relative percentages if given one. Now, going on to the numbers. So I give you the haploid genome. Haploid genome is half of a diploid organism's DNA. That equals 90. Do I not know how to do math? 21, no, it should be because 21 plus 24 is 50, right? 21 plus 24, no, that's 45. I don't know how to do math. This is why you never do math off the top of your head. So I am missing 4 percentages there. So so that should be 58%, right? 58, and that would make that 29, and that would make that 29. There we go. I was just checking to see if people were awake and actually listening to me. That's what I'm gonna say. Why isn't it 58%? Because I can't do math. That's why. Okay. So A is 29%. T is 29%. All right. Now 21 plus 29 equals 50. Yeah. All right. So I give you the haploid genome, which is 45,000. That's half of the diploid genome, which is in the cell. So the whole diploid genome is 90,000. And so there's a lot of ways you can actually solve this problem, but one way to do it is take the diploid, multiply it by, I'm going to solve for G and C here, multiply it by 0.21 for 21%, and you get the amount of G or C is 18,000, sorry, I'm doing half, 37,800. So that's the amount of G, and that's the amount, no, sorry, I'm all over the place. 18,900, I'm skipping a step. So that's the amount of G or the amount of C, right? So that's how much of G and C. So A and T, we can do the same thing. Take our 90,000, multiply by 0.29. And the amount of A and T we get is 26,100. So that's A or T. So that's one way to do it. You can do it via percentages. What you can also do is figure out how much G there is, figure out how much C there is. And if you combine G plus C, you get 37,800. What you can do from there is take the diploid genome, 90,000, subtracted by how much G and C there is, and you get 52,200. You could divide that by 2 to get the A and T. So just a couple of different ways to get it. But that is just Chargaff's rule in a nutshell right there. Some simple math if you have a calculator, because apparently I can't do math off the top of my head today. But yeah, so that's all we had to cover from Monday's lecture. So I'm gonna switch PowerPoints and go to today's lecture. All right, let me get presentation mode up. So in biochemistry, one of the techniques you do a lot is you manipulate DNA. And if you took molecular biology, you've probably already seen something like this before. But for us in a lab for biochemists, and for anybody who modifies DNA, one of the things you really need is an enzyme called a nuclease. What a nuclease does is that it will cut DNA. And throughout this class, we're going to be seeing enzymes a lot. So anytime you see ACE, you're going to know that's an enzyme. Like enzymes always have like ACE in the name. Now, a lot of the times, the name of the enzyme will tell you what it's doing. So nuclease means it's working on nucleotides, and endonuclease means it's going to cut in between the DNA, while an exonuclease cuts at the very end. So if this was my DNA strand, exo would cut at the very end here, endo would cut in the middle. Now, we know of two types of restriction enzymes. We have type 1, which cuts not at the recognition site. So what a type 1 does is that it will go and bind to some sequence of DNA and cut at some other random sequence. Well, it's not random. It'll cut so many spaces away from that, which isn't useful for us because when we're cutting DNA, we usually want to insert something or delete something. So we want to know exactly what we're cutting, not like, oh, we're cutting 1,000 ways from the sequence or 1,000 base pairs away from the sequence. So what we use in the lab is called a type 2. Type 2 will cut right at the recognition site. It will cut at the sequences it's looking for. Now, there are two different types two different cuts you can make. One will produce fragments that are called sticky ends. One will produce fragments that are called blunt ends, all right? And why they're called sticky and why they're called blunt, because if you look at where our enzymes are cutting, which is shown by this line, our DNA is gonna have overhangs. So we'll have G, then on the bottom we'll have C, T, T, A as one strand, and the other strand will have A, T, T, C with G. You have bases that are exposed, right? And so if you want to modify DNA, that is basically create a GMO, that's what a GMO is, it's modified DNA, you can put in your sequence that will base pair to this, and you'll form a, it'll make a much better connection. It's much easier to work with a sticky end, a endonuclease that produces a sticky end, because you have these bases that you can base pair to. While a blunt end, I'm not even going to draw the letters, but that's what a blunt end looks like. There is no overhang. There is nothing for your sequence that you're inserting to attach to. So if you're trying to put in a new sequence, you just kind of got to hope it will go into that gap and that it will insert the right way. By the right way, I mean not backwards. Because remember, DNA does have direction, which we read 5' to 3'. So it's always better to use a sticky end if you can. Sometimes you're forced to use a blunt end. Here on this table, we have a bunch of enzyme names, and we have a bunch of sequences that they cut. The arrow shows where they cut, and the bacteria where we get them from. So we find these endonucleases in bacteria because that is their way to protect against any foreign invaders. Now, I'm not going to ask you to memorize that table because that is a waste of your time. There are websites where you can look that up. When you buy these enzymes, it'll say right in the bottle where they cut. So don't worry about that. Just know what an endonuclease is, what are two types, which one's useful, what's the difference between a sticky end and a blunt end, and how, if given a table like this, can you determine if an endonuclease is a sticky end or a blunt end. So we're going to do that. So let's say we have this sequence. Let's say in A, I mix that sequence with Taq 1. What will we get out? B, tell me which sequences will make sticky ends and which ones will make blunt ends. C, do we have any isoschismers on this table? An isoschismer recognizes the same sequence. So two enzymes recognize the same sequence. And a bonus question I thought of while I was going through that last stuff, D. Yeah. So in D, I said all of these endonucleases are found in bacteria. Why don't these endonucleases cut the host DNA? Why don't they cut the organism they are found in? So we will get started with these questions right now. As always, let me know if you have questions or if you think you have an answer, especially for D. D's a thinker. Let me know in the chat, but I'll give everybody a few minutes to work on that. D, is it because the DNA of the host is methylated and nucleases don't munch on methylated DNA? Yep. So for every organism that contains the restriction endonuclease, they have another enzyme called a methylase that will go and methylate that sequence. So the restriction endonuclease won't chop something that's been methylated. So that's the way that they prevent themselves from being chopped up. Okay, so let's take a look at Taq 1. Taq cuts at TCGA. All restriction endonucleases, they're always written as cutting 5' to 3'. So when searching for a sequence to cut, you have to read 5' to 3'. You cannot find that sequence going backwards and expect it to cut. So if I look at the top strand right here, 5' to 3', I'm looking for TCGA. So we make a cut right here. And I look for TCGA going the other direction on the bottom strand. And I have it right here, TCGA. So if you notice, TCGA on one strand, on the reverse strand, it is AGCT, or 5' to 3' TCGA. So restriction endonucleases, for the most part, always cut palindromes. A palindrome is a word the same backwards and forwards. So the classic example is race car. Race car is the same backwards and forwards. Here, when we say backwards and forwards, we're talking about one strand versus another. So you look at the complement strands, and if it's the same 5' to 3', we call that a palindrome. So here, our cut's going to be like that. One strand's going to be ACGT, and it's going to be on the bottom here, TGCAGC. So we have an overhang. Our other strand is CGAATC, and you're going to get GATT. So again, we have some overhang here. So that will be a sticky end because we have those bases that are not hydrogen bonding to anything. And when you're looking at a blunt end versus sticky end, you're really just seeing, do I cut in the middle or not? So here for ALUI, you're cutting right in the middle. It's equal, 2 on each side. So that's a blunt end. Anything that's not cutting like directly in the center of something will create a sticky end. Here, Eco R2, that will also be a blunt end because it's kind of cutting in the middle again of something. So blunt, blunt, sticky, blunt, sticky, 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 blunt, sticky, sticky, sticky. So all you're asking yourself, sticky versus blunt end, is when I make a cut, do I have the same number of nucleotides on each side? If yes, that's going to be a blunt end cut. If no, that's going to be a sticky end cut. And last, which one are isoschismers? That would be HPA2 and MSP1. They both recognize CCGG. So here we're cutting at the same spot. It's not necessarily 100% that isoschismers always cut the same spot. It's just that they recognize the same sequence. That's what makes an isoschismer an isoschismer. Any questions? Could I explain A one more time? Sure. So in A, what you're doing is first you want to find your enzyme TAC1. TAC1 recognizes the sequence TCGA. This arrow means I cut between the T and the C. So what you do with your DNA is you start the 5' end, and you go and you look for the sequence TCGA. So here it is, TCGA. I'm cutting between the T and the C. Then on the other strand down here, I'm also looking 5' to 3' for TCGA. So TCGA, cut between the T and the C. And the fragments that I wrote over here are just what would come off when you make those two cuts. So imagine that like this box or this sequence was connected and you took scissors and just cut that piece of paper like that. What would you have left over? And that's what I wrote, and that's a sticky end because your piece of paper would have overhang to it. All right, any more questions about that? All right, let me clear this. I want to show you a resource you can use for this. So I'm going to stop sharing and go to my internet browser. Yeah, all right. So there's a whole database online that has restriction enzymes. And the site rather is called Rebase. So to look it up, you just Google Rebase restriction enzyme, and you'll get it, or the actual URL is rebase.neb.com. So what you want to do is go to enzymes. And it's going to list every single restriction enzyme that we know. So each one of these links is not a restriction enzyme. It's a list of restriction enzymes. So it's saying the restriction enzyme from AAA all the way to AAL1. And the methyltransferases to go with those restriction enzymes. So here are all the restriction enzymes you could potentially use in your experiment if you wanted to. Now, you can't buy them all. This is just all that we know. So if we want to use, let me see if I can find it off the top of my head, EcoR. So EcoR is made from E. coli, and it's well known. So EcoR, I did not get the correct list, I guess. Anyways, we can use this. So here's a bunch of ecos, and what you can see is supplier. Well, let's look at this whole list, right? So recognition sequence, what it's known to bind to, and if it's well studied, what it cuts, and then where you can buy it from, and if there's any isoschismers available out there. So this could be a resource for any of you who are doing projects and want to just look up, I have a genome and I want to cut it. What restriction endonucleases could I use? Or I found a restriction endonuclease, which one, or what does it do? Let me look it up. So that's just an online resource for you. So I'm gonna go back to my PowerPoint now. Okay. So the next thing we're gonna talk about is how to sequence DNA. And we're going to start with the old school method and then talk about the 21st century methods. So the old school method was around in about the 70s, 60s, 70s when this was really developed by Frederick Sanger rather. And it's called the Chain-Terminator method. And this method is actually really clever. So what you do is that you take your DNA, right, and you separate it. So you get your DNA, just a single strand, and then you separate that strand by heating it. So you break the double helix. Then what you do is you add DNTPs or DDNTPs. So what is a DNTP, just for those of you who are not sure? So DNA, basically, that's more or less what the D stands for. The N means ATCNG. It means all types of triphosphates. So when you see a DNTP in a solution, that means I have all 4 of my triphosphates available. So what does a DDNTP mean? To understand that, we have to draw the structure again of our base. Well, I actually forgot they have it right here. So here is DNA. Normally, DNA has an OH at this 3' position. Because if it has an OH there, we can connect another phosphate to that oxygen and continue growing our DNA. But we don't have that. Oh, this takes forever to undo. There we go. Instead, we have just a hydrogen there, which means we cannot connect any other triphosphates to this sugar. That's why it's called a terminator. As soon as we end this DDNTP to our growing polypeptide chain, we stop. It can no longer grow. And so the idea is you add a lot of this, the unlabeled DNTP, and few terminator DDNTP that is labeled with like something to make it phosphorylate under light. And what you do is you give each nucleotide a different color. So here, C is blue, A is green, T is red, G is yellow. And you just run this experiment a lot of times, a lot of times, where you just keep synthesizing. And if you do it enough, this terminator will be added at a bunch of different positions. And then you can separate the DNA by length and determine, okay, which colors am I seeing when I separate these by length. So in this sequence, I'm seeing blue, blue, green, red, blue, yellow, red, red, yellow, green. So I know my sequence is CC, ATC, G, TT, GA based on the colors. And this chain terminator sequence is actually still used today in labs. It's how I used to figure out my DNA sequences back when I was in grad school just a decade ago. So even though it was discovered a long time ago, it's actually still in use because it's relatively cheap. So are there any questions about the idea of the chain terminator sequence for determining how the sequence of DNA? Some sequences have the same color. How can you tell the difference between which is which? So each nucleotide, so the only thing that's colored is the last nucleotide that's being added, because that's our terminal nucleotide, right? So C will always be blue. And all these sequences that you're seeing here, it's the same sequence. So imagine that we have this really long DNA sequence, and we're using this method to figure out what each nucleotide is. Well, maybe the first time you run it, only one nucleotide is added. Your terminator is added first. So you know, okay, my terminator was added first. So my first nucleotide was C. Then you do a second run. And maybe this time, the terminator is just the second nucleotide added. So you look at the marker there, it's blue. So you know, oh, my second nucleotide's a C. Maybe the third time you run it, was this 1, 2, 3, 4, 5, 6, 7. Maybe the third time you run it, the seventh nucleotide added is red, because that's where the terminator was added. So now you know, okay, it was not until the seventh nucleotide that my terminator was added. So I know that my sequence has a T at the seventh position. So if you were doing this, you would know, okay, first position's a C, second position's a C, third, fourth, fifth, sixth, I don't know yet, I haven't got those results, seventh is a T. And the idea is you run this enough times, you run this sequencing enough times that a terminator by chance would be added at all these positions in different runs. You can see what nucleotide was added last at that run and figure out what sequence is. That's the chain terminator sequencing method. Does that make a little more sense about what's going on here? Okay. Yeah. So that is still used today in labs. Generally, what you do is you pay someone to do that for you. You don't do that experiment yourself. There's very advanced labs that do that. And you give them like $30 and say, sequence my DNA, please. And what you get out is a chart that looks like this. So when you do the chain sequencing method, you might have to wait like 24 hours, sometimes faster, sometimes longer. And you'll get an email back, or if you want, you can probably get a print, something delivered. But you get an email back that looks exactly like this. This is your sequence. You can see each color here. So this is saying, you know, when we ran this experiment, for example, at position 110, we always saw the signal for G because there's no other like color really there. So we are fairly certain that this is a G. Like at position 139, you can see there's some other peaks there. They're not really that big, but you can say like, I'm 98% certain that's a T. There's 2% uncertainty there. And what you'll see when you run the chain sequencing method is that the farther you go out, the more and more questionable it is until like hundreds of nucleotides out. All 4 peaks will be like basically the same because you have no confidence because like a terminator was like never added there. So the chain sequencing method is usually only good for a couple hundred nucleotides before you can't get anything out. But let's take a look at how you might actually use this information in a real experiment. So let's say I was sequencing a human protein. I was sequencing the DNA of that protein. In position from 130 to 150, my sequence should have been what's written there. Do I actually see that sequence? If not, what would I say it happened then? So looking at this chart, can you determine if we have the sequence there that I want? If not, what would you call that change? And B, you can kind of skip B. B is just looking through that and finding the cut sites, but we don't need to do that. It's not there. There's a C to a G. Yeah, so at position 143, we have a G there. We were expecting a C. So it should have gone TTTC. It went TTTG. That is called a point mutation. We have a point mutation, or more vaguely, a mutation, but that is a point mutation. So this is something that still goes on today. You get this back and you manually check it to see, okay, does this have the sequence I think it does? Is there a mutation there or not? And you have to verify it by hand. Is the sequence I'm working with what I think it is? So this is a common technique, and it's all based on science, 50 years old now. Right? So let's talk about how to sequence things faster, like the human genome, because you basically can't do the Chain-Terminator sequence for the human genome because we have billions of base pairs, and that will do like 100 at a time, a couple hundred. And so that would take forever. Now we can sequence an entire human genome for like $1,000 in a day or two. So if you wanted to, you could get your genome sequenced if you want to put down $1,000. So let's take a look at the different types of sequencing out there. The first is called pyro sequencing or 454 sequencing. And so what you do is if you have like DNA that you want to sequence, you will attach these DNA fragments to plastic beads. So you take your DNA, you can chop it up a little bit, connect it to these beads right here. Then what you do is you add, in this case, you add 1 nucleotide at a time. So what's happening is that I'm adding only T nucleotides. Now I'm adding a primer to get started in polymerase 1. And if there are any A's at the bottom, so it has to be like at the very bottom. So at the very bottom of your sequence, if there's any A's, T will be added. So in this case, we know that AA was our first 2 DNA sequences because 2 T's were added across from it. And what happens in pyro sequencing is that you wash one type of nucleotide over, you hit it with a laser, and then you measure how much fluorescence it's coming back. And the technology is so advanced that you can tell the fluorescence signal of 1 molecule versus 2 molecules. So you can tell if there was 1 T added or 2 T's added, 3 T's, so on and so forth. So you do that, then you wash away all the nucleotides that didn't bind and add your next nucleotide. In this case, we add A. We let the A interact with the DNA and see if any stick. So we shoot it with a laser and we get no signal back, which means, oh, so no A stuck. So there is not a T next. Okay, wash all the A's away. Next nucleotide, add G's. Okay, shoot it with a laser. Did any G's stick? Yes, we got one signal back, which means, oh, okay, so there's one C. So right now our sequence goes A, C. Wash all the G's away, do the C, see if any C's stick. They didn't. Okay, go back to the start with T. You basically just repeat that process over and over and over again until you have your sequence totally done. And this is roughly 1,000 times faster than the previous method we just talked about. So doing something like this, we can really speed up the process and sequence like whole genomes if we wanted to. So are there any questions on how pyrosequencing or /454 works? If not, I will move on to the next type of next-gen sequencing. That's Illumina. Illumina starts with more or less the same idea. You take your DNA fragment, you put it on, in this case, a glass plate, and you amplify it to have millions of the same molecule. The difference between Illumina and pyrosequencing is, well, there's two main differences. So one, you add all 4 nucleotides at the same time. So here we just add all A, T, C, and G into the mix. You just dump them all in. And the other difference is only one nucleotide at a time will ever connect, will ever bind. So let's say we have the same sequence that goes AAC. We add all 4 nucleotides. No matter what, only one T will ever bind. We shoot it with a laser, we get fluorescence back, and we see, oh, we saw the T fluorescence, so T must have been added. Wash everything away, add nucleotides, do it again. We see another T sequence, so it must go TT, so on and so forth. So it's similar to 454, but a little different. How they can add only one nucleotide at a time, that is secret company information, so they won't tell people, but that is how they do it. And so here you can read 3 billion nucleotides with one run. So you just chop up your genome, right? And you just read, and then you can put it back together after the fact from your short little reads and get a whole sequence, a whole genome if you wanted to. So any questions about our next gen sequencing techniques? Okay, so what do you do? Oh, so we do have a question. So the purpose of this sequencing is fast reading. The purpose is to do it fast and to do large genomes. So for the Chain-Terminator sequence, that works, and it's fine if you want to do a couple hundred residues, and it's cheap to do. So that's why you would do this. If you want to, or let's say you're studying like a person, right? And they have some kind of new disease and you want to study them, you would sequence their whole genome. And to do that, you need next gen. What do you mean by wash away? Do you really have to flush them every time? Yeah, you just kind of like, so you more or less wash over the your DNA sequence of nucleotides, then you have to get everything out of the system and reset. Does sound like a workout, but it's, I mean, a machine does it. So you as a researcher don't really do anything other than run the machine and read it. So it's pretty simple on your end. I'm a little confused about the diagram. I can briefly go over that again, sure. So this gray is the DNA you want to sequence. All these colors are different nucleotides. So T is green, and the key is over here, right? T is green, red is C, yellow is A, blue is G. This purple thing is our polymerase. That's what's connecting new DNA. This thing, this long black line's a primer, is just to get us started. And this is a glass plate. So you add all 4 nucleotides to your sample. You let your DNA polymerase work, and your DNA polymerase will only attach 1 nucleotide at a time. This lightning bolt is a laser. You shoot a laser at your sample. These nucleotides are developed to fluoresce light back when they're hit with a laser. And this is all done with a machine. Like you wouldn't physically look at it. A machine would record this. And so the laser hits, and you get a green light back, which means I attached a T nucleotide. And then you repeat. I add the next round of nucleotides. I let 1 attach. I use a laser, see what light comes off. I get green again. That means a T was added. Next, add more nucleotides. Let polymerase work. See what was added, shoot it with a laser. I get blue this time, which means a G was added, which means the nucleotides so far I've added were TTG. That means the sequence I'm trying to look at is AAC. Because if I know what's added, I know what's on the other strand, because that's how DNA works. That make a little more sense? All right. So again, as shown here on the bottom, this will work from like 30 to 300 nucleotides. But I've been saying this, this is for genomes. So how do we do that? Well, here I have a question where you don't actually have to do it. I'll show you what's going on here. But basically, after you do a bunch of short reads, you would get something back like this, where it just says, okay, 1 read, we got this sequence. 1 read, we got that sequence. 1 read, we got that sequence. And you have to put the pieces back together to determine what the entire DNA was. Now I say it's really a computer. You give this to a computer and a computer will tell you what it is. But let me show you basically what's going on. So imagine we took this long DNA strand and we just randomly chopped it up into different size parts, right? We went through these parts through Illumina sequencing and we got these reads back. And I want to know, okay, what is the sequence of this? It's basically like a puzzle. So let's say, okay, this is kind of what the computer's doing more or less, is that it's going to take the first sequence and just say, okay, our first sequence is T, C, G, C, A, C, G. So that's our first sequence. Does anything match up with this? Well, our second sequence kind of does. Our second sequence has a TCG, and before that, we have ATTA. So that's our second sequence. Does anything now match up with these 2 sequences? Well, our third sequence kind of does, because it has a CACG, and that matches with sequence 1. So I'm going to line that up. CACG, TCC. So all you're doing is you're lining up complementary segments of your DNA. Because remember, you kind of more or less chopped this up randomly. You chopped up many of the same DNA strands. So you need to have overlap to put the pieces back together. And so now you're asking, okay, this was that. So my puzzle is going like that. Do I have any overlap to any of the pieces I have up there yet? Yeah. Piece 4 has an overlap. So piece 4 is ACGTCCA. So that's piece 4. Now do I have any overlaps? Well, so I'm going to cheat here because I already did it this afternoon. So 5, 6, 7, 8. Piece 8 has some overlaps. So it has CCA. Here I have CCA. So CCA, TATG. That was 8. Then we have number 6 4, 5, 6. Number 6, you see where does that match up? Where does number 6 match up? ATG, ATG, TTGG, and then last number 7, we have TGGTGAA. And then after everything's lined up, you just read left and right to see what was my original sequence. So it's starting from here because that's the most left, most left nucleotide. Your sequence, the whole sequence is A, T, T, A, T, C, G. Then it goes C, A, C, G, T, C, C, A, T, A, T, G, T, T, G, G. Oh no, I'm running out of space. So on and so forth. Basically, you just start here and you move from left to right to figure out your completed sequence. And that is how you put the pieces back together. Do you have to read the way they are listed? It should always be the way they are listed, right? Because if we go back on our previous thing, let me get the arrow. You can set this experiment up in such a way that like the strands, like you can say this is like always 5 prime. But like I said, a computer is going to figure that out. No one's going to sit by hand and get like a million pieces and try to put that back together. A computer can do that like no problem whatsoever. They're very good at doing that. So any final questions about our overview of DNA sequencing here, as that is all the time we have. All right, so if you do, feel free to email me. Otherwise, I'll put our homework up. And just as a reminder, Monday's a holiday, so don't come here at 2 o'clock. I won't be here, but hopefully I'll see you on Friday. Take care, everybody. Have a good one.